I thought I'd begin with this reality show, Paternity Court. Now, has anyone seen this? Oh, we got one person nodding their head. Don't worry if you haven't seen it. You haven't missed that much. Okay. But anyway, the premise behind this reality show is that you get a couple that come in front of the judge and usually say that the husband believes that is not sure that their child is their child. Maybe she's having an affair or something like that. And then they each put their case, and at the end of the case, then the final decision, they do the DNA test, and all is revealed. All right, so the DNA never lies, and so you find out what's going on. And of course, this only works because we have very strict rules in society about who should mate with who. Okay, we have these barriers or societal barriers, and if you look at the Ten Commandments, 2 of them are dedicated to this issue. Okay, so you shall not commit. Number 6, you shall not commit adultery. Number 9, you shall not covet your neighbor's wife. So not only shouldn't you do it, you shouldn't even think about it. Okay, so they make absolutely sure. Now, we also extend this notion of barriers, reproductive barriers, or whatever you want to call it, outside the human domain. So if you look again in the Bible, you'll find this one. Thou shalt not let thy cattle gender with a diverse kind. Thou shalt not sow thy field with mingled seed. Okay, so again, there are strict rules as to what should breed with what. There should be barriers set between certain types of things. And this notion of these barriers and these different types relates to something that Ernst Mayr, the biologist, talked about, what he called typological thinking versus population thinking. So typological thinking is that we classify things into types. For example, here we have these true breeding breeds of cow, and we see the world as partitioned into these discrete categories, and that's applied to biological, to species or to breeds like this. Whereas population thinking is thinking more in terms of variation, in terms of individuals being different, not trying to just think in terms of these rigid categories and types. And of course, that's strongly related to the notion of barriers to reproduction. So the notion of types, you have these very strong barriers, whereas in the notion of population thinking, we think more in terms of frequencies of events and mutations and so forth. Now, although here the barriers are being created by humans, okay, the breeders have put in these barriers, but actually barriers like this, genetic barriers, exist quite prevalently in nature as well. So if you look at an ecosystem like this, there are plenty of barriers between different species. We see different species, they don't breed with each other, they don't cross with each other. And that's essential for an ecosystem, because if you imagine every species could cross with every other species, you wouldn't have a whole bunch of species. You have some sort of genetic soup, and ecosystems wouldn't exist. So barriers are fundamental to the way an ecosystem, for example, works. And this notion of barriers, or the notion of types, also extends to the way we then classify organisms. So here's a classification. This was provided by Richard Milne here. This is a classification of orchids genera based on their morphology. So this is classical taxonomy. This is the type of taxonomy that's most common. It's a more traditional taxonomy. You classify into types according to their appearance. But of course, just as we can use DNA to sort out paternity, we can also use DNA to sort out disputes over what should go where. Okay, so then if you sequence DNA, you end up with what one of these bars, these black bars here, these phylogenies, and it turns out, for example, the orchis, which was one genus based on the classical taxonomy, is now split into 3 different ones, some of which are more related to other genera. So just like in paternity court, DNA holds sway, and so we now change the classification. So now we have this. All right, so the DNA verdict is given, end of story. Okay, so what I want to do, though, is question some of this, because in one sense, this is making certain assumptions. One is that there's no hanky-panky going on between the species, all right? There's no, oh, I keep banging that, sorry. There's no gene flow going on between the species, no genetic exchange. But the other thing is it doesn't really explain how these branches are arising, what's causing barriers to form. Okay, so it's in a sense, it's replacing one type of typology with another typology. It's still typological thinking. It's just saying these types were the classical types. Now we've got DNA, will create a whole bunch of new types. And what I want to do in this talk is really start to think not in terms of types, but in terms of populations, but use DNA and DNA sequence information to help us think in terms of populations rather than these types, and think more about how these barriers are actually arising that carve up nature in the way that we see around us. So we want to talk about DNA variation and how we're going to do that. And one of the most common forms of DNA variation is what are called single nucleotide polymorphisms. These are variants. So here's a DNA sequence, and one position in the DNA is different in different individuals. So these are called SNPs. SNPs for short. These are positions in the genome where you find, for example, a G instead of an A or something like that. And if you were to sequence the genomes of everyone here, you'd find a whole bunch of SNPs that vary at different positions in the genome. Many of them are fixed, many of them don't vary, but some do. And that variation is why each of you looks different from each of the others. It's to do with these SNP variation. But what we want to do with SNP variation is think in terms of how do we measure that variation? How do we measure things like diversity using these SNPs? To give a comparison, if you think of temperature, all right, temperature is a measure we use all the time, but at rock bottom, temperature really tells us is how fast molecules are moving. Actually, if we drill down into temperature, it's built on a statistical notion, the notion of how fast, on average, things are jiggling about. High temperature, they jiggle fast. Low temperature, they jiggle slowly. And in the same way, what we'd like is a measure of DNA variation, a number that tells us what is the diversity in the population of these SNPs. What is the divergence between populations? What are the possible barriers that arise from that? And ideally, we want to measure a divergence that's going to be insensitive to what are called inbreeding or population crashes. Suppose, for example, you have a sudden crash in the population size. All of a sudden, the number of SNPs drops because there's not as much variation. And what we want is a measure that is not going to be sensitive to that type of fluctuation, because what we want to be able to do is just make comparisons without worrying about those effects. So what I'm going to try and do, and I hope this works, I'm going to try and explain this measure. All right, so bear with me because it's going to underlie the rest of the talk. And I thought it would be useful, instead of dealing with SNPs, I'm going to deal with socks. Okay, so here now is my population is a chest of drawers. Okay, and each of these drawers has lots of different socks, lots of socks in them. I'm only showing you 10 socks in this drawer, but you've got to imagine there's thousands of socks. That's our population of socks. And in this sock, we've got 50% are going to be pink and 50% are blue. And we can ask, how would we measure the diversity of socks in the drawer? All right, so one way of doing that is to imagine that you close your eyes, you go in, you get one sock, and you get another sock, and you ask, what is the probability that those two socks are different? That could give us an indication of how diverse that drawer is. So if we do that, the probability of pulling out a blue sock first time is 0.5, then a pink sock is 0.5, so that's 0.25. But I could do it the other way around. I could have pulled out a pink sock and then a blue sock. So the total number there is 0.5. So diversity, the measure of diversity, pi it's called, is 0.5. Okay, what if our sock frequency is different? Suppose there was 80% are pink. Now it's going to be 0.2 that I carry blue, then 0.8, or I could have taken 0.8 pink first and then 0.2. So that gives me a total of 0.32. So now the diversity is less. It's 0.32 instead of 0.5, which kind of makes sense. All right, the population is more homogeneous. If they're all blue socks, we do the same calculation, the diversity is 0. So this way of calculating diversity, which is to just imagine taking two socks from our drawer, is quite a good way of measuring that. Now, if I do that for my different drawers, I can do it my whole chest of drawers. All right. Suppose we have 100 chest of drawers, 100 drawers in my chest. Suppose 96 have a diversity of 0, 4 have a diversity of 0.5, that is a 50%, pink and blue. Then the average diversity within the drawers is going to be 4 of the ones that have 0.5 divided by 100. So the average diversity is 0.02, okay, because the other ones aren't contributing any diversity. Okay, so as you might have guessed, our socks are actually corresponding to these SNPs, all right? So what I'm calling pink and blue socks correspond to nucleotides, these positions in the DNA, which are variable. Some which vary, some which don't. And that we can equally measure the average, what's called the average nucleotide diversity in a population in exactly the same way with this measure of pi by multiplying these probabilities. Now, that's the diversity within a population, all right? That's like looking at the diversity in this room, assuming you're all one population. But now suppose we got another room with a different population, so another chest of drawers. Okay, now we've got 2 chests of drawers. We can need to ask, what is the what I'm going to call the divergence between the drawers? Now, since I'm not asking about how diverse they are within the drawer, I'm asking how different 2 drawers are. But we'll do exactly the same trick. I'm going to take my first sock from this chest. I'm going to take my second sock from this chest, from this drawer, from the same drawer. I can do the same calculation, all right? So in this case, it's quite easy. It's 0.5 times 0.5, exactly the same as the one I did before. Even though they're now different drawers, my divergence between the drawer, which is this sum, is 0.5, which is the same as my diversity within the drawer of 0.5. So diversity and divergence are here exactly the same, 0.5. Suppose they're all blue. Again, divergence between the drawers is 0 because I'm always going to pull out the same sock color. And similarly, diversity within the drawers is 0. But suppose now there is a difference in the frequency of yellow and blue socks. Suppose, for example, here we've got 0.8 blue, 80% blue, and over here we've got 80% pink. Now we're going to find that the calculation is 0.8 times 0.8 plus 0.2 times 0.2, which is 0.68. So now the diversity is higher. Sorry, the divergence is higher than the diversity. We've got more, even though here diversity is 0.32, the divergence between the drawers is different. Here's another example. Now, diversity within each drawer is 0, but divergence is 1. Okay, so it's a maximum divergence. Okay, so I can also calculate the average divergence between my drawers, my chests. If 96 have the same color in both chests, dxy is going to be 0. That's this divergence. If 4 of them have, say, 50% in both chests, dxy is going to be 0.5 for those drawers. So the average divergence, this dxy value, is going to be, again, 0.02, 2%. And that's exactly the same as the diversity within the chests, 0.02. Okay, so now we know what dxy is, and we know what pi is, divergence and diversity. Now suppose there's a catastrophe, and all of a sudden, all the socks randomly become fixed. Okay, this is like a population crash, but it happens randomly. So in other words, in each drawer, if I had 50-50 blue and red, blue and pink, by chance, they're all going to turn red, or they're all going to turn pink, or they're all going to turn blue. Okay, but we're going to do it randomly for the different drawers. What's going to happen? Well, remember there were four drawers that had 50-50. So on average, if this happens, sometimes they'll go, one quarter would go all pink, all blue. Another will go all pink, all pink, all blue, all blue, all blue, all pink. And so here's my divergence. It's dxy is 1, 0, 0, 1. So if I take the average, all right, the average is again 2%. Okay, whereas the diversity within the chests has gone to 0. So what does this mean? It means my population crash is not affecting my dxy, even though it's affecting the diversity within the chest, within the drawers. So that's what we wanted. We wanted to measure It's going to be insensitive to the fluctuations caused by population crashes or what are sometimes called selective sweeps. We've got this measure of dxy that's independent of that. And that means that we can use it now to study how things vary and the barriers between populations. Okay, so armed with this notion, let's now consider two interbreeding populations, 1 and 2. And if we measure their dxy and their pi, it's going to be identical to begin with because the populations have the same frequencies. And remember, if the same frequencies in each population, you'll have the same dxy and the same pi because there's no difference between them. Okay, now we put a barrier in, a red barrier. I'm going to take a drink here. So now they can no longer interbreed. What's going to happen? Well, initially, they're going to be the same, still stay the same. Their dxy and their pi is identical. But over time, divergence builds up. So dxy starts to increase. That's this blue arrow here. Whereas the pi, the diversity within, the orange, stays the same. Because pi is almost in equilibrium. Diversity is in equilibrium because you're gaining mutations through new mutations, but you're also losing them as things kind of become fixed, so that all the socks go one color versus the other. So pi is in kind of equilibrium. Diversity stays in equilibrium, but divergence between the populations increases because different colored socks now are being added to the drawers as these things are being separated. Now, let's suppose we divide these into subpopulations. Now we're starting to get something that looks like a tree, all right? We've got a root branch length here, which gives us the depth of the tree, this blue bit, and these orange bits are like the prongs of the tree, and the prongs are equal to this pi, this diversity, whereas the branch length is the difference between the dxy and the pi. It's the divergence that's built up while these populations were separated. All right, so that dxy is telling us how different have they become while they've been separated, or that extra bit of dxy, this root branch. Okay, so if we now take away that barrier, what's going to happen is all of them are going to exchange with each other, and this divergence between them is going to be lost because they're all going to revert back to the same value of diversity, because now everything's the same again, because they've all exchanged their genes, so their diversity is all the same. All right. So what does that mean? It means that if we can find these things with these deep divisions at the root, these root branches that are long, it tells us something about whether there's a barrier operating between our populations. Okay, that's an indication of a barrier. If we don't find it, it's an indication that there's a free-flowing gene. That's what this whole exercise has been about trying to convey. So we're going to now use that notion and apply it to a group of species. Okay, and the group that I want to apply it to are these snapdragons, Antirrhinums. Okay, so these grow in the Mediterranean, and there are a whole bunch of different. You also grow them in your garden, but those are domesticated horticultural varieties of snapdragons you grow in your garden. But these ones grow in the wild, and there are a whole bunch of different species. And they grow in different parts of Spain. But I'm going to choose to look at two very closely related ones throughout this talk. I'm just going to be talking about two of them. And these two, let me just say this taxonomy, this division of plants into these different species was done by this guy Rottmeiler in 1956. He did a sort of this is the Bible of Antirrhinum plant taxonomy. All right, but this is 1956. DNA, the double helix, was published 3 years before this. There was no DNA sequencing at this point. All right, this was all done on the appearance of the plants. We have this classification. So Rottmeiler gave all these species names, and previous people have had as well. And the two I want to concentrate on are here. These are called, this is Antirrhinum majus, var, pseudomajus. It's a variety of Antirrhinum majus, subspecies majus. Okay. And I've got another one, which is Antirrhinum majus, var, striatum. So they're very closely related. And the main difference between these two varieties All right. And what Rottmeiler was using to define them was their color, the color of their flowers. So here is striatum. It's essentially got a yellow flower with a magenta highlight. The bee, when it enters, bees go in and enter these flowers and pollinate them. And they enter at the point of highest contrast between this magenta and this yellow. That's the entry point. Now, this other species or variety has the almost complementary pattern. It has a yellow highlight with a magenta background. It's like two different road signs. One's red on yellow or magenta on yellow, and the other one's yellow on magenta. What they're doing, these road pines, is signposting to the bee where to go in and collect its nectar, collect her nectar. So what we did was we started to sample, sample populations from these different varieties. Okay, so this, I'm going to talk pretty much entirely about the work of a talented postdoc, Daniel Richardson. And what we did was plants were sampled from each of these locations. In each case, about 50 plants were sampled, and leaves were collected and crushed and DNA extracted. So we ended up with pools of DNA sequence, all right, from each of these. There were 18 of these populations. 9 of them were magenta and 9 of them were yellow. And then what Daniel did was he made a DXY tree, okay, to see, is there any evidence of barriers between these yellow ones and the magenta ones, okay? And what he found was there wasn't. Here's the DXY tree. You'll see that the yellow and the magenta are interspersed. It's not dividing them into yellow and magenta, okay? So as far as the DNA is concerned, as this DXY measure is concerned, yellow and magenta just kind of mingle with each other. So that means that Rottmeiler was wrong. Remember, DNA, we trust DNA. DNA gives us the verdict. Rottmeiler is wrong. This is correct. End of story. Okay. But there is an assumption here, which is that the entire genome is behaving in a completely uniform way. All right. Remember, this is based on the sequence of the entire genome. All right. And supposing, though, that different bits of the genome behave in different ways. Supposing, for example, that if you made a tree from this bit of the genome, you got a different result from making a tree from another part of the genome. That would tell us that maybe some regions are exchanging more than other regions. What you do when you do the whole genome is you get the average of all these trees. You put them all into one. But suppose there's diversity that we're missing. Supposing different parts of your genome have different properties and flow with different frequencies. So that's what we decided to do. What Daniel decided to do was start to scan the genome. But for trees, he'd make a tree here, tree here, tree here, thousands of trees now from the genome, and start to think about what they tell us. Now, in doing this, we thought, well, we'll forget flower color. Let's do this blind. Let's just ignore everything. Just think about the DNA. All right, we'll forget what these plants look like. We'll forget where they came from. We'll forget everything. We'll just think about the DNA. We're going to do this blind, so we're not going to be biased in any way. We'll just take the DNA of these 18 populations and just see what sort of trees they make as you go along the genome. Now we have to think about what sort of properties we're looking for. One of them is this that I've been talking about already, is this division depth. If this is a tree, what I call the root branch length, or the division depth, tells you how deeply divided is the tree at the base. And that's the thing we're most interested in, because that indicates there's some barrier going on. All right. So this one has a deep barrier, a deep tree. This has a shallow tree. So we think, so if we find something like this, we say, aha, this one has some sort of barrier to gene flow or gene exchange. We also can divide them into how they divide the group. So if you follow this division, this one's divided in a 2 and a 2. This one's divided at 3 as a 3 versus a 1. So we can talk about the type of division. So what Daniel did, well, just to give you an example, this, for example, remember I showed you the whole genome tree, that was a 17-to-1 division. Okay, that's 17 were grouped like this. But if you look at this division depth, which is quite small actually, there was only one tree separated from 17. So Daniel did this, and he did it for the 18 populations for 18,000 or so genomic windows. That means he went along 18,000 times and made 18,000 trees. Each of these were about 50,000 bases long. And what he got was that most of them give a 1 to 17 division. So if you look at this scale here, it's a logarithmic scale. So this here is 10 to the 4, or just over 10 to the 4. This is about 10 to the 3. So this is 10 times this. So most of them are in this column. All right, something like 90% give 1 to 17. But there are other divisions that you also get, which are at a more minor level. Like here, for example, is and we color coded them. But you can also ask, what about their root branch length? So what Daniel then did was he devised a way of saying, now I've got all these 18,000 trees. I'm going to start grouping them according to which trees look more similar to each other, have the same topology, the same arrangement. So now we're going to end up with forests of trees. Okay, so starting with 18,000 trees, I'm going to end up with a load of forests. And I can ask for each of those forests, what is the average root branch length or division depth for those forests? And that's what's shown here. This plots the forests color-coded in the same way as here. And there we can see the size of each forest. These are all full of different trees. They're full of trees, but they look similar to each other. And we look at the division depth. And you see, we're looking out for ones that have a deep division. And we see that there's one here that's giving a 9 to 9 division, that's in turquoise, and one here giving a 7 to 11 division. So these are standing out as giving us deep divisions. They kind of imply that we're dealing with some sort of barrier. All right, so what do these divisions correspond to? Well, the 9 to 9 division, the turquoise one, if you look at, this is what the tree looks like. There's its division depth, divides them into these two groups, and this is where those populations are located. In Spain, this is Spain and France, and we call this the A partition. That's the turquoise one. And then there was one that was giving us a 7 to 11 division. All right, and that's only 0.01%. Did I say that this one is 0.5% of the genome? It's a very small fraction of the genome that's doing this. This one is even less, 0.01% of the genome, gives this 7-11 division. That divides the populations like this into what we call the B partition. So what bits of the genome are giving rise to these different partitions? Okay, we have a tiny fraction of the genome. Well, you can map these regions that are giving these trees. And these are the different chromosomes of the Snapdragon of Antirrhinum. And here are the distribution. You see each of these, what you find is there are 8 regions that are giving rise, that are responsible for these deeply divided trees. 8 regions of the genome. 7 of them, shown in turquoise here, are related to the A partition. That's the one that divides it in one way. And one of them, number 8, is divides it in the other way, in the 7-11. So now the question is, what are these genes? And what are they doing? What do these partitions mean? Well, I'm going to first concentrate on the turquoise partition, these 7. And when you look at it, he says, waiting, yeah. This partition, the A partition, that's related to 0.5% of the genome, corresponds to the flower color variation. We didn't use flower color to get at these partitions. We just looked for deeply divided trees. Okay, but funnily enough, we ended up with most of those being related to flower color. All right. So what does that mean? It means we've got a very bizarre result. Rottmeiler was correct for 0.5% of the genome. He wasn't wrong. He was 99.5% wrong. But of course, quantity isn't everything. Maybe this is a really interesting part of the genome, and maybe we should be aware of that. So it tells you the genome isn't behaving all in the same way. And what Rottmeiler was detecting when he was making his classification was this peculiar percentage, this tiny fraction of the genome. So what is this tiny fraction of the genome doing? Well, as you might suspect, what we found that that a lot of these genes, these turquoise genes, turned out to be genes controlling flower color. We looked at some of the genes controlling flower color that we knew about. There's a gene called FLA and a gene called SULF, and another gene called ROSEA and another one called eluta. And it turns out that they map exactly to where some of these islands that I was showing are located. So some of these locations contain flower color genes. They're the genes that are making the thing different, making one flower different from the other flower. But there are these other ones here, 1, 2, 5, and 7, that didn't correspond to any flower color gene that we knew of. Or maybe we thought, well, maybe that's just we haven't looked carefully enough. Maybe there is they are also affecting flower color, but we just haven't noticed. So what we then did was you make crosses between different plants, and you generate families, and then you look at the variation in the flower color, and you see whether your gene or your island relates to that variation. And what we found, for example, here, if you look at the bottom, we've ranked this population of this family into from lowest yellow to highest yellow. And then when we look at the markers that relate to this first island, we see that they're enriched. The white marker is enriched in the high yellow, and the other marker is enriched in the low yellow. So you'll see this difference in yellow is hard to see, okay, by eye, right? But that was done just by ranking them. So it's quite a subtle variation. It's not surprising it was missed, okay, because it's quite a subtle variation. But nevertheless, this island harbors a gene that affects the intensity of yellow. The same is true of this island. It turns out that also harbored a gene that affected the intensity of yellow. And in fact, all of the turquoise ones turned out to control yellow or magenta. They all control flower color. So what's happening here is all of these genes that are showing this peculiar behavior are related to the control of flower color. Okay, so now we have to ask what's going on. Okay, why? What is happening? Why is this tiny fraction of the genome behaving in this peculiar way? How do we understand that? And to explain that, I'm going to not talk about socks. I'm going to use a different type of analogy, but also related to our culture, which is this. Okay, so here are two pictures. All right, this one is of Edinburgh, taken by my good friend Justin Goodrich, and this one is of Norwich. And you'll see they both have a Starbucks. Okay, so there's no problem with Starbucks invading the world, right? There's no barrier to Starbucks, it seems. Everyone loves Starbucks. Doesn't matter where you are. Okay, there's a free exchange of the Starbucks chain. You can also get it in China, anywhere you are in the world, you'll find Starbucks. All right, so that's one type of cultural feature. Now let's take another one. Suppose you were going for a skiing holiday. Where would you go? Probably not Norwich. Okay, so there is in fact a ski slope in Norwich, but maybe you wouldn't choose it. Maybe Scotland would be a better option. So here we have cultural choices that relate to differences in the environment. Okay, so we kind of have a barrier between Norfolk and Scotland related to this type of climatic or environmental condition. But there are certain types of cultural traits that have nothing to do with the environment and yet are distinctive. Okay, I don't know whether you can think of some of them, but here's one. Driving on the left and driving on the right. Okay, there is nothing. These are the countries. The red is the drive on the right. The blue is you drive on the left. There is nothing about the climate that has anything to do with driving on the left or driving on the right. It's purely a barrier that arises because actually driving in the middle is not a good idea. Okay, the hybrid combination doesn't work. Each combination works well on its own, but the hybrid combination doesn't work. So then you have to ask yourself, what happens in places like Hong Kong, right? So Hong Kong they drive on the left because it used to be a British colony. China, they drive on mainland China, they drive on the right. So what happens when you drive from Hong Kong to China? It could be, potentially dangerous. So they came up with this design, which I quite like, this bridge. I don't think it was ever constructed, by the way, but I like the idea that you would kind of drive along here, then you would sweep over onto the other side and so forth. So this is a way of getting around what we call hybrid incompatibility, the fact that solutions that work well together on their own don't work well when you try and combine them, okay? So we have then 3 possibilities. We have free flow, Starbucks, okay? We have environmental related barriers, like the things that relate to the environment, environmental differences, and we have a hybrid incompatibility barrier, all right? So which is the flower color? All right. Well, if you look at flower colors, this, as I mentioned, they kind of both would work. Okay. You can see why this is kind of complementary. This is like driving on the left. You've got your magenta highlight with a yellow background, or you can have a yellow background with a magenta highlight. Both work absolutely fine. But maybe the combination doesn't work. In other words, we've got 2 solutions that have been found in nature, but maybe the combination doesn't work. And evidence for that comes from this, from what's called a hybrid zone, where in the Pyrenees, you'll find there's a road in which you'll find there's magenta flowers on one side and yellow on the other side. And there's about a kilometer stretch in which you find these, okay, these hybrids, different flower colors, as if they're, well, we know that they're crossing with each other. They're exchanging with each other. So then we wondered, maybe these have, according to this incompatibility idea, these should have lower fitness. They should leave fewer offspring. Okay, but how are we going to find that out? It's actually very difficult to measure offspring, both parents in a population. But remember our paternity court. Okay, we can use DNA to help us. We can, with DNA, if we sample parents and offspring, and we catch as many of the parents possible, as many of the offspring as possible, we can do paternity and maternity tests and see whether things are leaving more or less offspring. So what I'm going to do this, we had to do very intensive sampling across this region. So what I'm going to do is a fly through. We're going to start here on the magenta side, and then we're going to kind of go along the road through to the yellow, showing you 1 year where we sampled all these plants. So let's see if there you go. So this is the magenta side, right? We're starting to get some hybrids, pinks, oranges. Now it's where we're in the hybrid zone, right in the middle. Now we're coming out. Please. So who says that science isn't exciting? Come on. So we did our paternity tests. We got a load of, we did the SNPs, and we started now to be using these SNPs to determine who was the parent of who. So then through the generations, through each year, we have to do this. All right, each generation, you see which plants you get, you relate them to their parents. These lines indicate descent. All right, you have to calculate with quite a few markers to be sure who is the parent of who. All right, so this is what we've done. Been working on this, for more than 15 years. This is what we call a long-term project. We still haven't published the results on this. We're trying. And what was the basic finding? Well, we found in this plot, what you're plotting is fitness in the vertical axis, how many offspring you leave. And then on this horizontal axis, we've got two one is the magenta, how much magenta you have, and then we have yellow, how much yellow you have, with the spread of magenta and the spread of yellow. Now the parental types, the yellow one here has got lots of yellow but very restricted magenta, and it has high fitness. The other one, at the other corner, we have magenta, the magenta type, which has lots of magenta and very restricted yellow, and that also has high fitness, indicated by, it has lots of progeny, indicated by the height of this column. But when you look at the hybrid ones, the ones that have the other combinations, all right, different combinations of yellow and magenta, they have lower fitness. So they have reduced fitness. So that fits this idea that they just don't function as well together when you make the hybrid combinations. So what's happening then is the reason that we have just this tiny little fraction of the genome, all right, that's showing these peaks here that come back with these regions that show this deep division, is because if you think about it, well, you know, not think about it. The fact it seems to be the case that 99.5% of the genome, sadly, is like Starbucks. Okay, that is, there is no barrier, that things can just flow freely. But there's a few, and this color in particular is one instance. Occasionally, you'll get something that follows the rules of hybrid incompatibility. That is, that the two different groups find a different solution to the same problem, but an equivalent solution. All right, but that doesn't work well when it's mixed. And that's quite a rare thing. Most of the times you'll find something that actually works well and can work well in both cases. But in some cases, very rarely, you'll find these hybrid incompatible phenotypes or adaptations. And that's what we're seeing with these flower color. And if you think about it, those are precisely the genes, also the things that might create incompatibilities between species, because we think of different species as being, in a way, different solutions to the same problem. Okay, if you think about it, it's not that one species is better than another species. They tend to be different ways of surviving and reproducing in an environment. So they kind of each is a different solution, but maybe the combination doesn't work very well. So it's kind of the same logic. But here we're seeing it in just a tiny fraction of the genome. It's kind of zeroed in on the genes that are telling us this very interesting type of behavior. Well, there's another, you might be wondering about this little lonely 8 over here, what it's doing, okay? And at first we thought, damn, our theory doesn't work, you know, that was so beautiful, there's one exception, what are we going to do? But it turned out, actually, this turned out to be interesting as well, because we found that this one was related not to flower color, but to plant height. Okay, this carries a plant height gene. This is again a family segregating for a cross between plants from different carrying different alleles. And what we found was that this gene, which we call MAX, relates to height. So B2, the mean of the B2 genotype or the B2 allele is lower than the mean height of the plants carrying the B1 allele. And plant height one of the things that plant height might be related to are things like adaptation to altitude. If you look, okay, so the B2 is short, it gives you a short allele, gives a short height. The B1 gives a tall height. So B1 is black here, and B2 are the white populations. If you look at the background, the whiteness indicates the altitude. And you can see vaguely there's an association between the being short and being higher, but it's for sure it's not 100% association. All right. In fact, if you did the statistics, you wouldn't find it was particularly significant. But it's a working hypothesis that this is related to maybe to this ecological difference between different environments, but that because height is controlled by many genes, and because it doesn't have this incompatibility issue, you can have other genes that compensate for the height, that also control the height, so that the barrier is blurred. It's not as strong as the barrier with the color, which is why we're not seeing a very strong correlation between height and this allele. But this is a working hypothesis. We don't know if this is true. We need to do more experiments to figure that out. Okay, so now our exceptional islands are due to a whole bunch of genes. Most are collect flower color, that are due to hybrid incompatibility, with this max maybe related to height and eco-adaptation to environment, or eco-adaptive cause of the barrier. In other words, these ones are like driving on the left and the right, and this one is like Scotland versus the skiing in Scotland versus Norwich. Okay, so to summarize then, how do we think of this as happening? So this is our DXY measure here. And imagine we had an ancestral species, and it split into two. One, for some reason, stumbled on the yellow combination, that found the yellow solution to attracting its pollinators. The other one stumbled on the magenta solution. And then they came into contact at some point. And most of the genes flowed. They were the Starbucks genes. Most just flowed between the populations. That was 99.5% of them. But a few were either eco-adaptive, as in maybe the max gene, that is, they were adapted to different environments. Maybe these guys lived in slightly different environments, or they were hybrid incompatible, as in the flower color genes, and that created barriers and prevented them from exchanging. So, going back to paternity court, the point is there isn't a single verdict with this story, all right, because the genome has many stories to tell. Depends on where you look. If you looked at the flower color, you'd see the classical taxonomy story. If you looked at height, you'd see something to do with eco-adaptation. If you looked at the others, you'd see a signal to do with flowing genes. So we can't really come away with a single typological idea. The DNA is not a type. Okay, you shouldn't think of DNA as defining a type, or at this level of analysis, you need to think of the genome as much more as a mosaic than in terms of a single type. And we're starting to think, as we're applying this the same tree method to other species, we're finding this isn't just a feature of the plants that I've been talking about, these very closely related varieties. It also extends to other species. So this notion of different of typology, of thinking of things as being this very discrete categories doesn't really apply. We need to think about how different bits of the genome are behaving. And it turns out that many of these species can intercross, and so that there can be flow of information between them. So when you look at your ecosystem, then to a first approximation, you can think of it as different types, like the classical taxonomists. And to some extent, the DNA will support that, right? But when we dig deeper, we see that if you want to understand how they became different, all right, or if you're dealing with closely related species, this notion of types breaks down, and you're dealing with different parts of the genome behaving in different ways, and barriers being created. And those barriers, those genes that create those barriers, are the bonds that actually ultimately lead to this distinction that you see. All right, so these barriers that are clearly here now and that give you that diversity are born not by thinking in terms of types, but by thinking in terms of this much more fluid picture of the genome. And on that note, I'd like to finish, but I'd just like to thank the people who were involved in this work. As I mentioned, Daniel already, but there are many other people involved in various components of looking at the genetics and the genetic control that I haven't had time to mention individually. We've also collaborated with the group in Toulouse, who are very important in terms of collecting this material that I was talking about from France and Spain. And we collaborated with Yang Biao Zhu, who in Beijing, their group did all the sequencing. And also population geneticists Nick Barton and David Field, and they were very important for trying to understand this pedigree analysis that I was showing you. Those lines between the different that paternity-maternity test actually is quite a complicated thing to do, and these guys were brilliant in terms of allowing us to get that done. So on that note, I'd like to finish. Thank you.